Hello everyone, welcome to my YouTube channel. Today we are going to see about morphology of bacteria. Okay, so the bacteria will measure used in microns. The unit of measurement used in bacteriology is micron, that is symbolized by mu. Okay, so the limit of resolution with the unaided eye is about 200 microns. So the size of the bacteria varies from 1 to 200 microns. So below 200 microns we'll be not able to see with the naked eye. So that's why we need microscope to see the bacteria. Okay, so then coming to shape of bacteria, basically we have cocci and bacilli. Cocci are oval or spherical cells. So this cocci we divided into, like we can categorize it into diplococci, that is the cocci arranged in pairs. Cocci in chains we usually call it as streptococci, and cocci in cluster we call it as staphylococci. So coming to bacilli, bacilli are rod-shaped or box-shaped cells. So bacilli have peculiar arrangement. So cocobacilli, where the length of the bacteria is same as the width. Okay, so example is Brucella species. Streptobacilli, bacilli arranged in chains. Example is Streptobacilli. Chinese letter pattern, or we say it as cuneiform arrangement. Okay, so here the bacteria are arranged in angle to each other. It will be appear as I, V or L alphabetical letters. Okay, so example is Corynebacterium diphtheriae. Comma shaped bacteria, that is curve shape appearance. Example is Vibrio cholerae, and you have special category of bacteria, we call it as spirochetes, which is in spiral form. Example is spirulum. Okay, so these are the bacilli morphology, that we have cocobacilli, streptobacilli, cuneiform pattern, that is Chinese letter arrangement, then comma shaped and spirilla. Okay, coming to bacterial anatomy, in this video we are going to see about the cell wall. Okay, so cell wall is the outermost layer of the bacteria. So you can see here. Okay, here you can see capsule is outermost, but some bacteria are not capsulated. If you take capsule, in the exceptional case, cell wall is the outermost layer. So you have plasma membrane, mesosome, cytoplasm, then you have flagellum, plasmid, everything. So we are going to study each and every part of the bacterial cell. In this video, we are going to see about the bacterial cell wall. Okay, cell wall is the rigid structure which is like outer membrane for the bacterium, and it weighs about 20 to 25% of the dry weight of the cell. Means if you take, if you remove the content of water in the bacterial cell, so the cell wall weighs about 20 to 25%. So these are the functions of cell wall. So it accounts for shape of the cell. So it give the protection against the osmotic damages, and it give the rigidity. It takes part in cell division, and most important is it is target site for antibiotic. Okay, so we take antibiotics for the bacterial infection, right? So this antibiotic will act on the cell wall of the bacteria, and it is target site for lysozyme and bacteriophage also. Okay, so how we can demonstrate the cell wall? So we can do several experiments to demonstrate the cell wall. It may be plasmolysis, microdissection, or you react with the specific antibody, or mechanical rupture, or differential staining technique. Okay, so this is the difference between gram positive and gram negative cell wall. You can see here the purple color layer, that is peptidoglycan layer, is thick in gram positive, it is thin in gram negative. Okay, and if you observe the outer membrane, porine is present in gram negative. Okay, and you have this protein structure which is rigid in gram negative. Okay, and you have lipopolysaccharide abundantly in gram negative, which is absent in gram positive. So this difference will make us to help in the identification. So we categorize